Thank you very much to the 10 people that stayed to the end of the conference. It's funny that Stacy, even when she's not here, she's taking time of my talks. She have this ability to overcome me always. The only thing is that she wasn't funny as usual, so I'm more relaxed. She's amazing. She's top scientist, and I admire her work. And by saying that, hi, if somebody don't know me, how many of you were yesterday in the talk? How much overlap I have? Good. I don't see anything, but good. So I tried, since I gave a talk yesterday, I tried to change part of that, not too much. So I'm sorry for the people that were yesterday and saw part of the things, but I'm trying to show the same path or things that I believe, but in a different way. So if somebody doesn't know me, I'm professor of the Technion, which is the Technology Institute of Israel. I also have a company. Altogether, we're around 90 scientists that working on this project that I'm showing you, which is mainly cannabis. My lab is also doing algae work, like plant biology in general. And the fact that this is my first slide, meaning that they didn't put the right, the newest version that I gave him. But we will manage that also. It's fine. So let me see. Yeah, no, I'm fine. Okay, so I will go that way. When I established my lab 8 years ago, I started to work with, I have a PhD in molecular and plant biology, and I did postdoc in cancer research. And when I established my lab, I want to create kind of multidisciplinary lab that will work on a plants that affecting cancer. One of the plants that were there outside, mainly by work by Manuel Guzman and Christine Sanchez, was this plant. And we started to work with that, and we got really good results. And going further 7 years, first of all, most of my work and my students working on this plant, but we create a multidisciplinary lab that doing everything from extraction to analyzing the molecules, understanding how they affect. And the reason for that, it's not that I chose that. This is my license from the Israeli government to do a cannabis research. This is my name, Professor Meiri David, my faculty of biology. If there is somebody here that don't know to read Hebrew, I'm translating. Even though you know the Bible and everything, you should. But so this is my name, Professor Meiri. This is the number of the license, R for research, 0001, okay? So we have Rafi Meshulam that in '65 started to work with cannabis, didn't ask for license, just went and take cannabis. In 2007, the cannabis moved to the healthcare. 2014, I was the first one to apply for a license, so no collaboration. I have a license to work in room 401, and that's it. If I'm going to the microscope in the first floor, I'm a criminal, and I'm a criminal, okay? But for that, I didn't have any choice. You can't send it to the chemistry department to check it. You can't use your friend LCMS to do metabolomics. Everything needs to be in-house. And this is the reason my lab became a lab that actually had to do everything. I have a lot of criticism in-house about that I'm doing everything and multidisciplinary, but it's not my fault, this plant fault, okay? He multidisciplinary, it's multidisciplinary, right? So cannabis, as you all know, I'm professor at the Technology Institute of Israel, and this is how it looks like. I never succeed to work with these kind of things. So cannabis, and I gave that as an example yesterday, you know, People don't understand that cannabis is actually not really growing in the nature. Like, as a plant biology, there is no reason for this plant to create 20, 30, 35% in its flowers of cannabinoids. We have many plants that produce cannabinoids. They will produce 0.1, 0.2. This is lipophilic, very sticky molecules. When the pollen, there is a male and a female. The wind is pollinated by the wind. The pollen is flying and is attaching, sticking by this molecule to the flower. But it needs 0.1, 0.2. The reason we have such an amount that because people did breeding for thousands of years with this plant and create something like we did with dogs, right? These are wolf, and this is, a wolf dog, very similar, but how do the hell we reach this one? This is cannabis, to my opinion, okay? I'm teaching genetics in the medical school, and I'm teaching mutations, and I said, yeah, mutation changing, and I'm showing this and said, how the hell we reach that one, okay? So this is cannabis, and cannabis harboring a lot of these molecules, phytocannabinoids. And phytocannabinoids were discovered by Rafi Meshulam, and I won't need to say any word about that, but they're affecting our endocannabinoid system. And again, I spoke about that yesterday. The endocannabinoid system is way wider than I think 90% of the audience here, and it's not a lot, knows, okay? I'm sure that, Sulek and other people understand it. But many people think that, two receptors, CB1, CB2, 3 molecules, anandamide, 2-AG. I'm teaching in Israel. In order to be physicians that prescribe cannabis, you need to pass a course. Also pharmacies or nurses. And I'm teaching in these courses most of the lessons there. But after me, there is the head of the Ministry of Health that's responsible to cannabis. And I'm teaching 3 hours the pharmacists about the endocannabinoids, where they're expressing, the enzymes, the breaking them, and 90-some endocannabinoids, 160 phytocannabinoids. The head of the Ministry of Health that's responsible to cannabis come after me, said, Daddy, thank you very much, always amazing talk. There is a difference between what's happened in the lab and in reality. In the lab, Daddy told you what happened. In reality, there are 2 cannabinoids and 2 receptors. Don't spoil, don't spoil the physician, think there is more than 2 cannabinoids, right? This is an extremely, extremely complicated system. Every cell in our body harboring the endocannabinoid system. My lab is working around 35 receptors that are participating in the endocannabinoid system. We can analyze in the blood samples around 90-some endocannabinoids. Altogether, we think there is around 130, 140. There is something like 50, which we call them endocannabinoids, like very complicated system that balancing, keeping homeostasis on our body, okay? Actually, it's a communication between 2 cells. One cell sends a signal to other cells, create appetite, reduce pain, affect sleep, increase insulin, and so on and so on. It's just communication. One part is the ligand, the endocannabinoid. The other part is protein called receptors, capture that, send a signal. Cannabis was just unlucky to produce similar compounds, and we create a poodle, right? Because we need it. But in the end, it's just producing similar compound and binding these receptors and signaling that our body knows how to do. Our problem that we don't know enough about the endocannabinoid system and the signaling, and more important, we don't know which molecules from the cannabis is mimicking the process that we don't know what the endocannabinoid is doing. So it's going and disappearing, but in the last 7 years I saw, I think, more than 500 startup companies that claim that they know which doing what. It takes me 30 seconds to break them apart. Like, we don't know. Mostly, we're starting to understand a little bit CBD, THC, CBG, not to the depth that we need. And one of the problems that cannabis is not one plant. In my lab only, we have more than 1,000 different chemovars of cannabis. I'm the dream of every stoner. Unfortunately, I'm not a user. I'm trying to work now with psilocybin mushroom. Maybe this will, be effective for me. So there is tons of chemovars. Each one of them contain different combination of molecules. And we don't know to match the right flower to the right bride, okay? And after 25 years of marriage, I can talk about matching and flowers to the right bride, and what's happened to the right bride after 24 years, but this is a different story. We don't know how to match the flowers, and this is actually the problem, to connect all these dots. Endocannabinoids, receptors affecting the endocannabinoids, the cannabinoids, the chemovars. We are far from understanding that. So my talk today was how to match that, but I'm run out of time, so I'm not sure we will reach there. So I want to show you how me and my lab and my team trying to approach this question in 3 different ways. One way is when we have a question on something that we have zero knowledge, okay? Cancer, how, so I will skip that. So this is saying that, no. Here, no, yes, no. Questions? Okay. You born with English. We are starting to learn in grade 4, I think, English. And the first lesson called yes, no questions. So it's a joke in Hebrew, yes, no questions. So where we start? We started, first of all, creating tools to do this research, and I will skip it. We created tools to analyze all the phytocannabinoids. We can analyze their 11 different families, altogether around 160 phytocannabinoids. We can do that also to the endocannabinoid system, taking a blood samples, tissue, or biopsy. We can analyze around, as I said, around 100, a little bit more endocannabinoid. We're participating in almost 20 clinical trials when we're getting blood samples, tissue, to start to get a profile of these endocannabinoids. I'm working with Stacy, getting from her. We're trying to put our hands. I won't touch this at all. The point is, I want to show in the 17 minutes that I have, 3 approaches how we succeed, how we're trying to match the right flower, the right combination to the right indication. So first thing, if we don't know anything, we don't have any information from patients how cannabis is affecting real cancer, okay, for an example. So we want to test how cannabis is affecting leukemia, breast cancer, colon cancer. So how we do that? We're doing that. We can grow cells in plate like that. We can grow cells. This is cells in the plate. This is one cell. The dark side inside is a nucleus. If somebody don't know how cells look in a microscope. So we grow cells in a plate like that contain between 3 to 10 million cells, depend on the types of the cells. We can grow cells also in plates that have many wells. We have equipment to run these fast and screen these wells and get an input. I won't go into that just because this frightening clock is going down very fast. Literally every second, it's 3 seconds here. So we can screen it very fast. So we ask how it's affecting leukemia. We know that cannabis is affecting the immune system. We have a lot of data from Crohn's, colitis disease, autoimmune disease. We know that cannabis is affecting the immune system. So we're asking how cannabis is affecting the cancer of the immune system, leukemia. So early, a PhD student in my lab started to work with TLL. This is a specific type of leukemia, and he took a TLL cell that's growing in the cell, screen about that around 200 different cannabis strains. So every color is a different strain, and we see viability. So if the line is lower, the meaning that more cells are dying. So you see that in 2 micrograms per ml, most of the cannabis strains didn't affect the cells at all, but there was one buster that killed these cells like hell. So we said, okay, leukemia, it's a given name to many different types of diseases. There are TLL and CLL and BLL, and I won't go into that. Let's see how it's affecting in the long range. So we took this extract number 12, and they joke in the lab that Ellie screened around 300, but extract 12 was the good one, so he can stop at 13. So we screened that on 10 different leukemias, and we saw that in 50% of the leukemias, the cannabis is not affecting, but in 50% of them, it's killed like very efficient. We did a genetic screening, and we saw that every time there is specific mutation called Notch1 mutation, the cannabis is killing the cell. Every time that there is no mutation, different mutation, it's not affecting the cells. In TLL, 50%, almost 60% of the patient will harbor Notch1 mutation. Notch1 is a protein sitting on the membrane, and in general, if I'm flattening that, he's feeling that there is another cell near him and that there is something called contact inhibition. If there is cells around you, you won't proliferate and create another cells. But cancer cells, which become crazy, one of the things that happen, they're losing this contact inhibition, proliferate and create a tumor, right? So Notch1 in the normal place, he's standing, feeling, think this is 3-dimensional, there is many proteins, feeling the cells around them, and they are fine. If there is no cell, it's vacant in time, there is other protein can cleave that, Notch1 become transcription factor going to the nucleus and create other proteins to proliferate and defend on the cells. Never mind the biology about that. But also, he sent a new Notch1 back to the membrane because he was cleaved. You need to create a new one. So, seeing that extract number 12 is very effective, we went to a mice model. We created tumors. You see a CT of the mice with a tumor in his back, right? We create this tumor. We treat them with extract number 12. The black line on the left is the size of the tumors that they're treating with a vehicle, with a kind of placebo, not placebo, with a control. You see the size is growing exponentially. The gray line is the size of the tumor treated with extract number 12. It didn't affect them. In the middle is the stat of the mice. The right size is the size of the tumor after 3-week treatment. They are shrinking. Because this is leukemia, we did a bone marrow implantation. We doubled the lifespan of these mice. We did actually 5 different models of a mice model to this, including primary cells directly from the patient to the mice. It's worked very efficiently, okay? Now, the $100 million question, from all of these compounds, from these extract, do we need all of them? The entourage effect, do we need 1, 3, 5? My slogan to my students, I want to know the minimum compounds that doing maximum effect. It may be 1, it may be 5, okay? So how are we doing that? We're taking this extract and we're cutting into pieces. We're doing fractions, okay? We said, okay, there is 100 compounds. We had the equipment to separate them to 25, 25, 25, 25. We're going back to the cells or to the mice, and you can see the vehicle, the control is not killing the cells. Extract number 12 is very efficient. And then the 4th fraction. And Ellie was more lucky than smart that fraction number 2 worked as good as the whole extract, already eliminating 75% of the other cannabinoids. These are the biochemistry behind it, not important. We keep doing that, and in the end, we succeed to isolate 3 phytocannabinoids that if you isolate them, make them synthetic or whatever, 100 purified, you put them together, you get exactly the same effect as the whole extract. But every combination, this is the left side, the low bar left side, every combination of one of them or two of them are not working as good as the three of them together. We did a lot of work, a lot of biochemistry to understand the mechanism of action. I don't have time to show that, but the bottom line, we understand today exactly why you need the three of them. Actually, we did what we called synergism testing, and two of them, CBD and CBDV, are additive. You can replace one of them by increase the other. So we don't need the both of them. We can reduce it to two molecules. In the extract, we need the both of them because it's additive. But if I'm taking purified, it's enough too. But the other one, a unique cannabinoid, we call them 3318, he is a derivative of CBD, very different structure. And CBD and him binding two different receptors. We are binding CB2 and TRPV1, activating proteins called ceramide in the cell that creating ER stress, elevating three different proteins, ATF4, CHOP, and CHAC1. CHAC1 is going to the Golgi and blocking NOTCH1 to go back mature to the membrane. Again, I don't know how many physicians or biologists are here in the crowd, so I'm trying not to go to deep biology. I just want to transfer the idea that we know exactly why, when there is a NOTCH1 mutation, cannabis will kill the cells. It's not anymore Rick Simpson oil. We think maybe we know exactly why, when there is no mutation, it won't affect the cells. When there is mutation, why these cells committing suicide, to the bottom of the mechanism of action. And from that point, I can convince any hematologist to try to use it on his patient, to go to real clinical trial, to prove it on humans, and that's what we're doing. Most important, you need to understand that if now when we're saying, yes, cannabis can kill cancer, I'm just showing you that from 300 different extracts, just one of them was effective, and from this one is affecting just 50% of the patient. So what is the stat of success of a patient that going to a dispensary and pick the right cannabis to kill the right cancer? It's a lottery. And it's not that I don't believe that cannabis can be effective to cancer, but I don't believe that it's killing every type of cancer and every type of cannabis. I think there is a lot of work to match the right flower to the right disease. So we open a company and we raise money and we are going clinical trial with that. And there is two pathways. One pathway to take extract number 12 and treat the patient. And actually in Israel, if you're an oncology patient, you immediately get prescription for cannabis in order to reduce pain, nausea, increase appetite. So actually, the patient today, if you're an oncology patient, you get the prescription, you can go and take extract number 12. It's on the shelf. So I don't need to start safety. I can go directly to the patient, work with the oncologist to just match it. So quite fast, in a year and a half, I can be after POC, proof of concept, that this extract working on this patient, checking NOTCH1 and everything. The other direction is pharma direction. If somebody wants to donate to me $200 million, I will do that also. We're doing that, but I'm looking for the donation. So this was when we know nothing. We're starting what cannabis will fit to Alzheimer, to leukemia. We don't have knowledge. But there is another way. If I have previous knowledge, in 2015, I got $5 million to do the Israeli database on patient. Having the ability to know all the compounds, I applied and said, I'm the only one in the world that actually knows what the patient is getting, not just, white widow and black forest. I can look on the molecules. So we got $5 million to create the database on all the products that patient can get. There was just 8 authorized growers, around 80 to 90 products, and we had agreement that every time that they're picking a batch or creating a new product, it will come to our lab to analyze it. On the other hand, we follow up on the patient and create a database, a shitty one, unfortunately. Almost worth nothing, and I can give a good talk about 2 hours how to spend $5 million. But it gave me a tool to start to understand where really cannabis is affecting, what are the strains that are better. So now I don't need to start to screen 300 strains. If I want to work on sleep disorder, I can choose, I will skip that, I can choose one strain that I know that is the best for the patient and start from there. And again, I run off the time, I will say we create a screening on the mice. It's very funny, and sorry for the people that heard that yesterday. We are following on 3 different parts of sleep disorder. When a mice is sleeping when he's light and is awake when it's dark. So when you're turning on the light, the mice will arrange his bed. It will take around 15 minutes to arrange his bed. It's a very funny thing because there is a big difference between male and female. The female will arrange her bed around 15 minutes. The male will arrange his bed around 7 minutes. Literally, after I heard that, I want to do an experiment. I want to see how much time take the male to arrange his bed when the female is around. Between 10 to 11 minutes, okay? He's not doing the full exercise, but he's better, okay? So we separate them and we follow up how much time it takes him to arrange his bed, how much time he's sleeping, the duration, and also a mice is lifting his head around every 2 minutes to look around. There is no mice and no cat around. So we are measuring these 3 parameters. One is giving you idea how fast or how tired he is. Second, how deep his sleep is. And the third one is the duration of sleep. So we're choosing, these are the strains that we chose. And we started with the strain that worked the best to the patient. So we already know that it's worked. This strain is working on humans. Now we're going backward to the mice to check what is doing this effect. So this is a strain that was very effective. The purple one is the control. Green, red, and blue are different batches of this strain that was very efficient by creating the mice to be very tired and go to sleep fast. Also increase the duration of the sleep. And again, the question, these are all the molecules in this strain. Which of the molecules is doing this effect? And the same methodology from here. We're doing a fractionation, cutting this to pieces. And I will skip that because I have just another 2 minutes and I want to show more things. We started to fraction, we cut out the THC, it's worked the same. We said, okay, there is also quite a lot of CBN and CBG. We cut the CBN out, it still worked the same. We said, okay, let's cut now the CBG and the other. We left with just 2 molecules, both of them unknown molecules, and these 2 molecules working as good as the whole extract. And this is when we are now, we know exactly which molecules are there. There are 2 unknown molecules, derivatives of THC and CBN, but never mind the details. We're calling them GTS, go to sleep 1 and go to sleep 2. Maybe we'll change the name, I'm not sure. But this is where we are, and from here, again, we have 2 paths to go. One of them is to take the extract, to cut it in here, to take out the THC, take out the CBN, left with the part of the extract, 0 THC, 0 CBN, but still effective of sleep. And I can tell you that a big population of people that have problem to sleep don't want to take cannabis because of the THC. My mother, my father, a lot of people. And this is a new product, 0 THC, 0 CBD, still extract with a lot of cannabinoids, but very efficient to put you asleep. Second, again, is a pharma way. Take these cannabinoids, go pharma direction, and so on. So this was second method. First method, if you don't know anything. Second method, if you have an idea on something, you can start with a strain that you know that is affecting one human, try to isolate and to find why it's effective. And the third one is more bioinformatics tool. So again, from the database we collected, we saw that there is no indication in Israel for migraine, but we saw that many patients, whether they are PTSD patient, cancer patient, also say that they are suffering from migraine and cannabis helped them. So we looked on just on these patients, around 500, and we sent this patient questionnaire for migraine. And we know that the endocannabinoid system is important in the role of creating and preventing migraine, so it makes sense that cannabis will be effective of that. So we collect data from this patient on the migraine, and we try to divide which strain are not helping or create the migraine to be more severe, and which strains are helping. On the strain that helping, because we know all the molecules, we try to look which molecules are appearing in all the strains that are there. We find 5 molecules that are unique molecules and not the major one that are appearing in most of the strains that reduce migraine. We looked on them, we gave them name, we started to look where they are. We analyzing them, we separate them, we understand the structure of each one of them, and now after we understand the structure, we're doing modeling about which one of them probably will bind the receptors that affect the migraine. I won't go into that. Oh, it's nice, now the time is going up, so I have lots of time. So I'm done in 15 minutes. 2 minutes. So we know the mechanism, we know. It's not, the mechanism of migraine in part is known. We know which receptors in the brain are responsible to migraine. There are treatments for that. So we did now modeling about these 5 molecules that we have, which one we think will bind these receptors and which one will fit to the receptors. After we find out which one of them, we isolated that and tested now in cell model and mice model that it's really binding, first of all, that it's really binding the receptors, and after we find it's binding the receptors, it's really affecting the activity of the receptors and reducing migraine. So we started with totally bioinformatics, from the knowledge that we had until we find the molecules, and now we prove that it's really working. After we find it, we can modulate these molecules, doing chemically changes. I know this crowd don't like it, but we're taking synthetically, modified it that it will actually work better. But more of that, trying to see if other cannabinoids can work together, again, bioinformatically, and we're improving the affinity and activity of these molecules 100 times more active than the one we really found in the beginning in the plant. So summarizing what I showed, I think that cannabis is harboring what we called polypharmacology, okay? And again, yesterday I had time to explain that. I won't go deeper to that. I think for people use plants for thousands of years as a medicine, but in the last 100 years, we know which plants affect what, and pharma company isolate molecules, and we call it single molecule, single target, create a medicine from that. And more than 50% of the medicine, where these are chemotherapy like Taxol, vincristine, where there are a medicine to reduce fever like aspirin, coming from plants. But it's single molecule, single compound, single target. In the last 20 years, we're starting to see that there are diseases that we need, in order to affect them, to attack them and to modulate them in more than one target, more than one area, okay, one direction. And we call it polypharmacology. We still in medicine doing that on medicine that each one of them was authorized as a medicine, and we're combining them. Cancer is a classic example. When we give more than one chemotherapy, we're giving kind of a cocktail, C plus T plus B, or plus biologic, okay? One is attacking the DNA, one is blocking the cytoskeleton, one is blocking the receptors, and we're treating the patient like that. If we think about plants, they are harboring inside of them polypharmacology. The physician 3,000, 4,000 years ago looks like that, and if you come with inflammation, he wouldn't give you one molecule. He will pick one flower from here, one root from here, one leaf from here, combine them together, and will give you something that he created as a polypharmacology. Maybe 80% of this is placebo, but also it's laying about a lot of data that's been acquired by these treatments. And I believe that the next step of complicated medicines that will overcome diseases that we can treat, like ALS, Alzheimer, types of cancer, and so on, will come from complicated treatment, and plants are harboring that inside. We already have this knowledge. Now we need to understand why it's working for that, okay? So the change that happened in the last 20 years is computers. Understanding different routes, understanding networks, today can be done in computers very efficient that 20 years ago it was almost impossible, and I think this is the next step. What I think cannabis did, cannabis is actually an orchestra of molecules that acting on different receptors in very complicated way. And I think if people think that Rafi Meshulam need to get a Nobel Prize because he found the cannabinoids, I think he need to get a Nobel Prize because he opened the gate for all of us to understand the complexity of plant biology, of polypharmacology. Cannabis give us the tools, open the mind of physician to treat with complex, to treat with plants, and gave us the tools, laboratories like mine, to start to do it even more on other plants. Not that I think that I have enough from cannabis. I think way after my retirement, there was a lot of work to do just with cannabis. But I think the message is way, way, way bigger than just the cannabis plant. This is, so what I think, actually, what I have today in my lab and my team, it's not a lab that's doing research on cannabis. It's actually kind of methodology, how to work with this complex plant, how to do extract, how to analyze active compound, how to run them on different diseases, define the specific effect, mechanism of action, preclinical, clinical, how to take something from its initial complex when we think we know that it's actually work on humans, and taking all the route up to clinical trials. So this is the amazing team that work under my supervision. They're doing the work when I'm traveling in the world, and I'm grateful for them, and thank you for listening.